Thanks, Rich, for that kind introduction. I am Stephen Skates. I'm at Massachusetts General Hospital and Harvard Medical School. I'm an associate professor and investigator on early detection of cancer. This talk is a sequence of trials for the early detection of ovarian cancer. It's our odyssey from 1985 to the current date, 2021. And I'm honored to have been invited by Karen and the Virtual Prevent Cancer Dialogue Committee to present to you on early detection of ovarian cancer. I want to acknowledge a large group of collaborators. These are the main people, but by no means the only people, and funding from multiple resources, both in the US and the UK. Enabled these studies. My declarations of interest, the biggest issue is that I'm an eternal optimist for early detection. And I think if there's any sway or bias, it's coming really from that. I'm also a co-investigator on UK CTOX, which is the main trial that I'll be presenting here. I've been a principal investigator on the Cancer Genetics Network ovarian cancer screening trial and a co-principal investigator on the GOG 199 trial. And there's been over 30 years commitment to ovarian cancer screening. And so that's where the conflicts really arise. But I am a co-developer of the risk of ovarian cancer algorithm with Ian Jacobs that has been licensed by our institutions to Abcodia, and I'm consultant for the following companies. However, none of these interests will impact my presentation today. The outline of the talk is that we'll talk about early CA-125 screening trials, a development from there on what we refer to as the risk of ovarian cancer algorithm based on longitudinal CA-125, where essentially each woman serves as her own control, and that's much more powerful than having a single threshold for CA-125. I'll talk about screening trials using ROCA, the design of the big trial and the mortality analysis in that trial, and where we're going in my vision for early detection of ovarian cancer from there. The first trial I was involved with was the Stockholm screening trial. Really, it's a Swedish-wide screening trial from over 6 years, from 1986 to 1992. I joined MGH in 1986, and my first project was on reviewing an application to the NCI for a small business innovation research grant that Centocor, who had licensed CA-125 from Dana-Farber, where Bob Knapp, co-discoverer of CA-125, was, along with Bob Bast. And they discovered it in the late 70s and managed to create it in the create an assay to it for it and apply it for second look operations in women, and it was FDA approved for that indication. And then they envisaged we could use it more broadly for early detection. And so they identified Sweden as a place where there was the highest incidence of ovarian cancer in the world, and managed to get this small business innovation research grant and screened with that 5,500 women over the age of 40. And if the CA-125 was greater than 35, they referred the woman to ultrasound. And in 5,500 women over roughly 3 years, they identified 6 postmenopausal cases of ovarian cancer, conducted 12 surgeries for suspected ovarian cancers. All of them were detected by CA-125. 2 out of 6, that's 1/3 in early stage. So encouraging. And then in parallel, there was the Royal London Hospital screening trial led by Ian Jacobs, and he had started a year earlier. He enrolled 22,000 women. They were postmenopausal, age over 45, and he did an initial prevalence screen on all 22,000 women, where his rule was if CA-125 was greater than 30, they were referred to ultrasound and a CA-125 every 3 months. And then he randomly selected 11,000 of those women to return for 3 more annual tests. That was the Royal London Hospital study. And 28 cases were detected within 1 year of the last test. So if we're thinking of doing an annual test, that's the relevant number. And 20 out of those 28 were detected by the screen. So that's a 71% sensitivity. And combined with the ultrasound, there was a 99.9% specificity, which was excellent. However, 6 out of the 28 were in early stage, and it was too low. So what can we do about that? What can we learn from those trials? And if you look at CA125 over time, what you see in the ovarian cancer cases is a rapidly rising CA125. And this is on a logarithmic scale. So 1 to 10 is the same as going from 10 to 100. So this represents a tumor doubling time. In contrast, in other diseases, benign gynecologic and non-gynecologic diseases, most of the women, when looked at over time, had a flat profile for CA125. And you could have, and especially with no disease, you could have high levels at times, but very constant, no rising over time, in contrast to most of the ovarian cancer cases. So that was the Stockholm trial. Similar distinction between a rapidly rising CA125 in the cases identified by the red dots here, numbers 1, 3, and 5. In fact, number 5 gives us our quintessential, what we think is of our paradigm, where we have a flat baseline and then ovarian cancer arises, and then it starts to rise very rapidly, much more strongly than the fluctuations that you see over time in the women that didn't have ovarian cancer, which was the green 3 women, 2, 4, and 6, that had varying levels of CA125, some of them very high, but over time there was this not the rapid rise nor the extent of the rise that we saw in the cases. And all of these, in the red cases of ovarian cancer, were prior to the clinical detection of the disease. So this is preclinical dynamics of CA125. And we came up with, Ian and I came up with the idea of this risk of ovarian cancer algorithm, where you have a regular CA125 test, and then you calculate the chance that they have this change point, this risk of, and use that risk or that change point, the probability of a risk, the probability of a change point, as a measure of the risk of having undetected ovarian cancer. And if it's normal, you return in the standard test interval of annual testing. If it's intermediate, you maybe repeat the CA125 in 3 months, and if it's elevated, you send them on to transvaginal sonography. And in that way, they have a baseline. You establish a baseline for each woman and look for significant changes above that baseline. And that way, we triage by risk, and essentially funnel those that are at highest risk for having a change point, that is having undetected ovarian cancer, into the most significant resources, which was the ultrasound. And we implemented that in a pilot trial of about 14,000 women, half screened, half not. And in the first 10,000 screens, we saw that about 2% ended up with elevated risk, either 1% of them by immediate elevation of CA125, or another 1% by an intermediate risk step, or a second intermediate risk step leading up to about 2% at elevated risk. And those women that are elevated risk went on to transvaginal sonography and repeat risk of ovarian cancer. And out of the roughly 200, about 10% had an abnormality on ultrasound, which required them, which led them to go to a referral to surgery. So out of those 22, 5 had ovarian cancer, and the encouraging feature of that was 3 out of the 5 were early stage, which is 60% instead of the usual 20%. So we had a 3-fold increase in early stage. Now, there were 17 false positives, but that gave you a positive predictive value of over 20%. And this was not enough to institute a national screening trial, a national screening program, but it did give us preliminary evidence for conducting a definitive trial of ovarian cancer screening with this approach. And at the time, there was a lot of negativity about ovarian cancer screening. So we had poor survival, or annual surveillance by CA-125 was ineffective, or we could not prevent the disease with CA-125 and ultrasound, or there were implications for screening women at high risk that were questionable, and that there was no effective screening in the PLCO study, for example, which was the US study that included, the O part included ovarian cancer screening. So nonetheless, we conducted multiple trials, and this gives you the timeline. This first trial was the pilot trial that was randomized 14,000 women over 6 years. That provided the preliminary data for the big UK trial, which is number 3 UK CTOX. In parallel, we also did a US risk of ovarian cancer algorithm trial in 2,500 high-risk women. And this was through multiple networks that was sponsored by NCI. UK CTOX, a UK collaborative trial of ovarian cancer screening, enrolled 200,000 women and ended up being conducted over 15 years. And half of them were randomized to screen and the other half to no screen. So we had a randomized comparison here in number 3. And this is what I'll focus on. There's also the MD Anderson cancer study that used this risk of ovarian cancer algorithm on 7,000 normal risk postmenopausal women. There was a US GOG gynecologic oncology group trial where the screening arm of that trial enrolled 1,600 high-risk women. And then there was a UK familial ovarian cancer screening study that enrolled 5,000 high-risk women. All of them published, all of them encouraging using this risk of ovarian cancer algorithm. In all of those ROCA trials, there was a significant increase in early stage cancer. And no other ovarian cancer screening trial had such an increase. All of them, all the other ones, used a single cutoff for CA-125, usually 35. In contrast, we individualized and personalized the test using ROCA and having each woman serve as her own control. This led to the definitive trial, the UK collaborative trial of ovarian cancer screening from 2001 to 2015. Ian Jacobs was the principal investigator, along with Usha Menon and 4 other investigators, including myself. So 200,000 postmenopausal women were randomized. It took us 4 years. We thought we could do it in 3, but it ended up taking 4. It was originally designed for 8 years of annual screening and 2 years of follow-up from 2001 to 2011. Turns out that the women who, there was 1 million women invited, 20% accepted, that's the 200,000. Those 200,000 were actually a healthier subgroup than the general population. And therefore, they had fewer cases of ovarian cancer in the control arm that we were monitoring. And so we realized that to get the power in this study that was originally intended, we had to expand the trial, and we added another 3 years of screening and another 1 year of follow-up. So in the end, instead of a 10-year trial, it was in fact a 14-year trial. Those women, 200,000, half of them were randomized to screening, half of those using the longitudinal algorithm, another 50,000 to annual ultrasound as a first-line test, and 100,000 to the control group where standard of care was given and no screening was done. So the results of that in 2014 were unfortunately not a significant difference, but you can see that the curves start to separate maybe around between 7 and 8 years. And these curves are the mortality curves. So this is how many women died cumulatively of ovarian cancer screening from the start of the trial at time 0 all the way through to 14 years of the trial. And you can see that the screened arm, which is the red, the multimodal screening arm, which included the ROCA algorithm and followed by transvaginal sonography, as a second-line test for those that were positive by ROCA, had fewer ovarian cancer deaths than the control arm, which is shown in the blue, C for control. However, this mortality difference by the Cox model had a p-value of 0.1. And you'll know from basic statistics that we want a p-value less than 0.05. So it was double what we needed to get for statistical significance. However, if you subdivided this timeline from 0 to 7 years and from 7 to 14 years, then you saw two different effects. There was an 8% reduction from 0 to 7. So you can see the red curve for most of it is underneath the blue curve, or at least for a large fraction, but not by much. And that had a very wide confidence interval. But if you looked at just the second half of the curve from 7 years through to 14 years, then there was a 23% reduction, and that had a confidence interval from 1% to 46%. And if you combine those and you looked at a 30-year screening program, these 7 years might have an 8%, but then the 7 to 14 years in the rest of the screening program for 23 years might have this 23% reduction. And when you then weighted average those out, then you get a 20% reduction. So that's what we might predict for a long-term screening program. But with the proviso that this p-value here wasn't quite significant. However, we did also look at excluding the prevalent cases. So if you look at the CA-125 over time for each individual case, what we found for most of the cases was these blue dots established a baseline for each woman, and this level was different for each woman. And then once they got ovarian cancer, then the CA-125 started to rise rapidly until they were detected on ultrasound, and then they had the surgery at the time of their detection of the disease. And what we can do is work out where that change point was, and if that change point, this is the change point from a flat baseline to a rapidly increasing CA-125, if that change point was after when they first got their screen, which is shown by the green line here, then these were not prevalent cases, they were incident cases, they were after the first line of screening. However, if the CA-125 was rapidly rising right from the word go, from the first screening test, as is the case here, and then we backtrack to where the change point is likely to be, we can see that most of the area where it probably was is to the left of the first screening test, and therefore this is a prevalent case. The disease was already likely there at the time of the first test. And so we excluded these because these cases, these prevalent cases, have much less chance for screening to have an effect because the disease is already there. And when we excluded them, we then found that the average mortality reduction was 20%, and this had a p-value of 0.02. That is, it was significant, less than 0.05. And this is the cumulative curves for the multimodal screening and the control arm, and you can see that there is indeed a bigger separation than there was when you included all the cases. So we did an additional, because of the mixed message there, we did an additional 5.5 years of follow-up. The data was closed in 20th of June last year. Data cleaning and analysis was finished in December. And then we submitted, reviewed, and published 2 days ago as of 12th of May this year. And what we were hoping is that, as in the prostate screening with PSA trial that was done in Europe, mortality reduction was marginal on the first publication in 2009, yet with another 3 years it was significant. And here it was marginal for UKC tox, and we were hoping in 5 years it would be significant. Unfortunately, I have to say it was not. So that was our odyssey with UKC tox. These are the people that did it. I'm right here at the back, but at the front is Usha Menon and Ian Jacobs, who led that trial. And then where are we going from here? Longitudinal algorithms, I think, still have a role here. but an annual testing for CA125 with CA125 leads to a 10% late-stage reduction, which is not sufficient to lead to mortality reduction. However, we are looking at other biomarkers through the NCI's Early Detection Research Network. We're coming up with algorithms that look at multiple markers over time. And we're also looking at uterine lavage as another source of a signal for the presence of tumor DNA in the uterine lavage and potentially in pap smears, and looking at serum protein biomarkers. And this is through the Early Detection Research Network. And I think that has great hope for early detection in normal risk women. And I want to say thank you to all the audience and to the Prevent Cancer Foundation. Thank you.